that's on. Why isn't this working? All right, I'm going to be immobilised. Okay, hope everyone had a good break. Probably the fact you've got a mid-semester test in an hour or two or tomorrow morning probably doesn't make you that cheerful about having had a break. Okay, so the last few weeks we've talked a lot about transcription. We've looked at transcription in bacteria, in eukaryotes, we've looked at RNA polymerases, we've looked at how bacteria genes are organised in operons, you've got regulators that can control the expression in response to different stimuli, and in eukaryotes we've looked at how there's transcription factors that are required for transcription to occur, and these can be general or specialised ones. And we've also looked at how histones can sort of hide DNA from these transcription factors and from the RNA polymerase. So the good news is that's it for transcription. Yay! Finally, you say. So we're now going to move on to sort of the next stage, and we're going to look at what happens to that messenger RNA that's now been transcribed. So today we're going to talk about messenger RNA processing. And so in the first half of the lecture I'm going to talk about messenger RNA processing in bacteria, and then the second half we're going to look at eukaryotes. So if we start off with bacteria, we know, I actually have a laser pointer that works now. Woo. So we know that RNA polymerase can transcribe either a single gene or a set of genes organised into an operon, and as that messenger RNA is being transcribed, before that transcript is complete, it's already being translated at the ribosome and turned into a protein. So this process of transcription and translation then are linked to each other and happening essentially almost simultaneously. That's it for the first half of the lecture. That's it for messenger RNAs in bacteria. Now for the second half of the lecture. It might take a little bit longer than the first half. So we already know in eukaryotes things are usually more complex, and that's true here. So transcription and translation, while they're coupled in bacteria, they're certainly not in eukaryotes, in part because they occur in completely different compartments of the cell. And so the messenger RNAs need to be moved out of the nucleus and then moved to the ribosome. Other complicating factors are that eukaryotic genes are interrupted by non-coding DNA, so-called introns, and these need to be, and so the messenger RNA needs to be spliced to remove these introns, and it also goes through some other processing that we'll talk about. So if we look at introns first, and hopefully has everyone heard of introns before? Yes, no, hands up. Okay. So, I didn't bring along my paper cutouts. And my pair of scissors. Oh well, no props today, sorry. Too disorganised after the mid-semester break. So introns are intragenic regions that are not translated. So they're in the DNA, but when that DNA gets transcribed into messenger RNA, those intervening sequences, those introns, are spliced out and removed from the messenger RNA. And so the regions that don't code for amino acids to be turned into proteins and are introns, the coding regions of the genes that actually encode what will eventually become a protein are known as exons. Not all eukaryotic genes have introns. Since I'm now leading the Yeast 2.0 project in Australia, I can tell you with some surety, which I couldn't have last year because I wouldn't have known last year, that only 5% of yeast genes have introns. And those that do usually have at the most 1 or 2 introns. Many other lower eukaryotes also have relatively few introns, but they all have at least some. And one other thing that we've found out is in this Yeast 2.0 project, we were trying to make a completely synthetic yeast. The original design was to remove all of the introns, and what we've actually now found out is that some of those introns are actually essential for cell growth and survival. So we've had to modify our original design as we discover these essential introns. When we get into high eukaryotes, most, probably nearly all genes have introns, and some have a ridiculously large number of introns. As far as I'm aware, this is the record holder. The human titin gene has 362 introns that have to be spliced out to make the mature messenger RNA. And some genes have introns that are up to 1 million base pairs in size in the human genome. So these aren't, often introns are just small, 20, 30, 50, 100 base pairs. They can sometimes be enormous, up to 1 million base pairs or more. And so the process that occurs then is this RNA splicing process. So here's our... I don't know if the recording will hear me as I walk over here. Actually, I've got a laser pointer, maybe I don't. I'll hang around here. So here's our gene on the chromosome. It consists of a series of exons with introns in the middle. That gets transcribed by RNA polymerase to make this RNA, which is an RNA with each of the exons, again separated by the introns. And then that RNA is processed or spliced to remove all of the introns and glue each exon to the next exon to make this final transcript, which is then what gets sent to the ribosome and translated to give a protein. How does the cell know where to splice things? Well, the splicing isn't random at all. Each gene, the splicing occurs at the same sites. And what we've found is across virtually all eukaryotes, the splicing signals are very uniform. So the first two bases of an intron, G and U, and the last two, A and G. So those are pretty much absolutely conserved. Then there are regions adjacent to those particular bases that are highly conserved but not identical. So I can't remember if I've shown you this sort of representation here before, but this shows a consensus sequence. So this is looking at the splicing sites for many, many, many genes and looking to see how well particular base pairs are conserved. And the conservation, the higher the conservation, the higher the height. And so what you can see here is that this is the almost absolutely conserved GU, and then around it are bases that are pretty well conserved but not as well conserved. This is the almost absolutely conserved AG, and then there's a region here which is somewhat conserved but not as well conserved as the AG. Now, if we take a particular gene and we do site-directed mutagenesis to change, let's say, arguably to change that G to a C, you'll very likely mess up the splicing. So if you mutate the splicing signal enough that it doesn't work anymore, so if you do something more drastic and say remove that AAG, I'm sure that wouldn't work then. And so when that happens is you get abnormal splicing where the right exons don't get joined to each other and you may not get the intron cut out. So what's the mechanism of this? How does this splicing reaction occur? And sorry, there's some chemistry here, but that's okay. I was a chemistry major, but that was, I can't even count that high anymore, 25 years ago, 24 years, some large number of years ago, I majored in chemistry, so my knowledge of chemistry now is essentially non-existent. There's an intermediate in this process of splicing, which looks like a lasso or a noose, or your textbook often calls it a lariat, which is a term that I wasn't personally familiar with until I had to teach myself splicing. But I guess it's another word for lasso. And because we're more, this is a cheerful unit, we'll show it as a lasso rather than as a noose. So there's a two-step, the reaction to remove the intron and glue the two exons together is a two-step process. In the first step, the 2' hydroxyl group of an adenosine nucleotide attacks this first bond here and breaks it and forms this circular intermediate then. And then the second process, the hydroxyl group connected to this guanosine residue here will then attack this and form a new bond there, joining the two exons together, and then this intron gets released as with this weird lasso loop structure in it. That's the phosphodiester bond.
Oh.
That probably says what I just said. This weird lasso-like intermediate, the RNA then gets released, and because it's got this weird structure that you never see anywhere else in the cell, the cell then realises that this should be taken away and degraded. So that lasso-like structure actually serves a secondary purpose, which it signals to the cell that this is now an unwanted intron that serves no other useful purpose, and the cell knows to break that down and then reuse the nucleotides. Oops, this is about to shut down on me. Let me get my ID card out. Give myself some more time. So, at this branch point here where this lasso forms, there's an A residue there with that hydroxyl group, which is doing that attacking. So, we've already looked and seen how there's conserved regions around the 5' and 3' splice sites of the intron. What about that A? Will any A work? Or is there some specific signal that tells the cell what A should be used for trying to make that lasso-like structure? And the short answer is yes, there's a specific signal. And while the 5' and 3' splice sites are very well conserved across all eukaryotes, this branch point around this A, the signal there is very species specific. So in yeast, that sequence is always UAC, UAAC. In high eukaryotes, the sequence is different and more variable. But the final A out of that is the A which then does that chemical attack on the 5' splice site. And so that, just to make that clearer, that A then there is that A there. How do cells do the splicing? Too many C's and S's and things in that sentence. So there's cellular machinery that's responsible for this. Splicing doesn't just happen by itself. There's complex machinery known as the spliceosome. And the spliceosome is a complex array of different things. So you've got your messenger RNA that's going to be spliced. You've got these 5 SNRPs, is how you pronounce it, in case you're wondering how to pronounce snRNPs. They're called SNRPs. These are small nuclear ribonuclear proteins. So these are proteins and RNA that function together to make these SNRPs. So there's 5 SNRPs, and then there's some additional proteins. And these 5 SNRPs are called U1, U2, U4, U5 and U6. And these are responsible for playing the crucial roles involved in splicing. So just to remind you, these SNRPs consist of both an RNA and proteins. Now the interesting thing is the RNA in them is often what's doing the recognition of where the splicing is to occur. And so if we look at the RNA in the U1 SNRP, it actually has sequence that's complementary to the 5' splice site. And we know from crystal structures and things like that that does directly base pair with the 5' splice site. I should say that you don't need to memorise the exact details of what every SNP is doing or anything like this, anything like that. Another SNP, the U6 SNP, has an RNA which has a complementary sequence to the 3'. It has a complementary sequence to the U1, the region of the U1 SNP RNA. And we know it also associates with this U2 SNP. And the U2 SNP has a region that is complementary in sequence to the branch point and can base pair there. And also has a region that's complementary to the 3' splice site. And the U2 SNP also interacts with this U5 SNP. So there's this complex series of interactions between the RNA in these SNPs and the messenger RNA itself, and between each other. So the spliceosome then consists of both proteins and RNA, and these have to be assembled in a particular stepwise cycle. I used to have a diagram of this in the lecture. I think I no longer do. It's insanely complex. It's in your textbook. I used to just show it up to sort of melt people's minds with its complexity, but I guess I got tired of that. And the cell can use control of the assembly of the spliceosome to control the degree of splicing that occurs, and that will actually affect levels of gene expression. This assembly of the spliceosome also requires energy in the form of ATP. Now we have a structure now of some of these SNPs. This is the structure of the U1 SNP. And what you'll notice, hopefully pretty rapidly, is that it's got this sort of ring-like structure. And this is actually a ring made of 7 proteins called SM proteins. And then the RNA itself in the SNP then fits in the middle here. One reason I show you this is actually, those of you who know Bridget Mabbott, part of her research is actually on this, is on relatives of these proteins that also are ring forming proteins involved in binding RNA. And so Bridget over the last few years has been making various rings that consist of different combinations. And so she's managed to make, I think, 6-member and 8-member and high number rings. And she's trying to build nanostructures where you can make nanotubes by getting rings then to sit on top of each other, with the eventual goal of trying to get this to be sort of a nanotechnological, useful machine where you can make cylinders from these ring-like proteins and then potentially functionalise. You know, they already have some function, they can bind RNA, but maybe you can give them other functions by functionalising their interiors or exteriors. Actually, I don't know why I just don't get Bridget to give this lecture. But there is also, in animals, there's a second spliceosome. And this recognises a rare variant type of intron. This rare variant intron has the same sort of thing occurs. It's got 5' and 3' splice sites. It's got a branch point that involves an A. They're highly conserved, but they're quite different from that in normal introns. And I should actually find out. I don't actually know why animals have the second spliceosome, or I don't know if anyone knows why. If there's any particular feature of this set of genes that requires differential control of splicing. And this different spliceosome also consists of SNPs, and it has RNA surrounded by these ring proteins, and so these other members of this spliceosome, like U11, performs the same sort of function as U1. So there are analogous SNPs in this, but they're different in sequence, and they recognise these different 5' splice sites, 5' and 3' splice sites and branch points. That's not all. SNPs can't SNPs by themselves can't do splicing. There are additional splicing, additional proteins, additional splicing factors that are required to help SNPs bind and carry out their function. So for instance, SLU7 is a protein that's required for correct 3' splicing site selection. If that protein is, if the gene encoding that protein is knocked out, then you get weird splicing where the branch point is about 30 nucleotides away from the normal branch point. And then, so I think to make relationships work, you need some level of commitment. You also need commitment to make splicing work. There's a whole set of proteins called commitment proteins, which are RNA binding proteins, and these are required to attract the U1 SNP in the first place to get splicing started. And this just shows the commitment proteins involved in yeast and compared in mammals, which is similar, but they're involved in sort of connecting the U1 SNP and making this sort of bridge across between the 5' and 3' splice sites, and then the rest of the SNPs can come up and correctly interact. And we have an animation. I haven't checked it this year. Hopefully it still works.
The following animation will describe the process of RNA splicing,
Let me pause it a
an
second, get the
important
volume
step
higher.
in creating the mRNA that is in process of RNA splicing, an important step in creating the mRNA that is involved in protein synthesis via the process of translation. Key factors in this process include RNA possessing introns and exons, and the spliceosome. Here we see an RNA molecule with a single intron. Several signals exist within the intron that are used in the splicing process. From the 5' end of the intron, these are GU, the A branch site, a pyrimidine-rich region, and the 3' AG. The GU and AG sequences define the beginning and end of the intron. Splicing is mediated by the spliceosome, which consists of several protein RNA complexes. The first step involves two complexes that bind near the GU sequence. The RNA is then looped, and 3 other protein RNA complexes bind. This final complex then undergoes a conformational change. Introns are non-coding RNA sequences that must be removed before translation. The process of removing the intron is called splicing. The intron is then cleaved at the 5' GU sequence and forms a lariat at the A branch site. The 3' end of the intron is next cleaved at the AG sequence, and the 2 exons are ligated together. As the spliced mRNA is released from the spliceosome, the intron debranches and is then degraded.
And so that little tiny little molecule that was coming up and breaking apart, that was ATP providing energy at different steps of that process to allow it to occur. Now, each gene does not necessarily have a single set of one doesn't have only one possible splicing outcome. Many, maybe most eukaryotic genes are subject to what we call alternative splicing. And in alternative splicing, then you have one gene that can make several, possibly many, in some cases hundreds or thousands, of different messenger RNAs that are different because they've been spliced in different areas. And so that's when when the Human Genome Sequencing Project was starting, or was running, there was this betting competition among scientists where you could try and guess the number of genes in the human genome. And people had done a lot of EST sequencing, which was sequencing just the cDNAs coming off from messenger RNAs. And based off that, the popular estimate of the number of cells in the human genome was about 100,000. And in that betting competition, there were, I didn't actually bet because I didn't know anything about human things. I only knew about bacteria, so I didn't have an informed opinion. But many of the bets ranged up to 200,000 genes or more. I can't actually tell you the final number of genes in the human genome because it's still a debatable question, but that number is on the order of 23 to 25,000. And so the reason everyone got it so wrong was because many, many, many genes in the human genome are making these alternative splicing to give alternative different transcripts from the same gene. And this alternative splicing can have profound effects on the protein that's encoded by the gene. And so this shows one example here. So here's a gene with 4 exons shown here in blue. And this can be alternatively spliced such that we either have this last exon here having this yellow and red region, which gives it a signal that that protein should be secreted, or it can be alternatively spliced such that that yellow and red region is spliced off into an intron, and instead this exon is fused to these green and red exons here. And if that happens, that has a different targeting sequence that targets the protein then not to be secreted, but instead to be located in the membrane. And so this is an example how one gene then can make 2 proteins which are targeted to entirely different parts of the cell. And so often these alternatively spliced products can either have different targeting, they can either have different activity, so they can either be active or totally inactive, and that can have profound consequences in the cell. While I'm here, just you'll see this in the next several slides, where you see these sort of lines like this here. That indicates 2 exons being spliced together. And so when we're looking at alternative splicing patterns, you can sort of show them with these angled lines like that. So here's just another example here. And it shows 2 different, on the above the line and below the line here, it shows 2 different alternative splicing patterns for this particular gene. So in this case, you can either have the genes expressed from a promoter here, which expresses this, and then this A exon gets spliced to B, spliced to C, spliced to D. This end bit of D gets spliced to E, E spliced to F, the end of F spliced to G, and then that's the end of the transcript. And that makes then, when fully processed, that makes this RNA here. Alternatively, you've got a variant here where a different promoter starts the gene from the B exon, and the B exon gets spliced not to the C exon, but instead to the D exon. But then there's another alternative splicing here that splices off part of the D exon and splices to E. The E exon gets spliced to the second half of the F exon, and then that's the end of that gene, and then the G and H exons get spliced to each other to form an entirely different gene. Is that true? Or did I mess it up? Oh, it doesn't. it's done that. I don't, actually, that last bit isn't quite right. Doesn't matter. Different splicing patterns then can lead to entirely different final RNAs that then get translated. And as I've already said, some genes can have just one splicing pattern, some can have two, some can have many, some have thousands of different alternative splicing events. And so this is somewhat counterintuitive because it goes against the central dogma of one gene gives you one RNA, gives you one protein. In some ways it's a challenge to that because you can, in this case it's one gene due to splicing is giving up to thousands of different RNAs that can give thousands of different proteins. Just to show an example of how this can be significant, alternative splicing particularly matters to you if you're a Drosophila or fruit fly because it totally affects what sex you are. So sex determination in fruit flies is all determined through alternative splicing. And you have a set of genes that then give either female or male transcripts. And so this particular gene, SXL, the default splicing pattern gives all of these exons spliced together, and that gives, when translated, when spliced together, it gives this final processed messenger RNA here, and in exon 3 there's a stop codon. And so this protein then is not fully translated in males and isn't functional. But in females, you get this alternative splicing pattern where exon 1 gets spliced to 2, but exon 3 doesn't get spliced in, 2 gets fused to 4, and so on. And because it misses this particular exon here with a stop codon, you get a functional protein. The function of that protein is to interfere or affect the splicing of this second protein here called TRA. And so normally the default pathway here is exon 1 gets spliced to exon 2, and exon 3 and 4 get spliced together to make this protein. But in females, this alternatively spliced version of SXL affects the splicing such that 1 gets spliced to 3 and 2 doesn't get included. That protein then affects the splicing of another gene, DSX. So in males, DSX has these 6 exons spliced together to make this protein, but in females, DSXY, the protein encoded by the female version of the TRA transcript, interferes with splicing such that 1, 2, 3, and 4 get spliced together, and then 4 has a stop codon and a poly A tail, so it doesn't. It only makes a protein with these 4 exons here, and then this particular protein signals a whole cascade of hormonal and other things that lead to that fruit fly being female, whereas the default pathway here then leads to the fruit fly being male. So sex determination then in Drosophila is entirely dependent on these alternative splicing events. And maybe it's interesting that sex differentiation in fruit flies has to do with commitment. So these versions of TRA, either TRA or as you get in males or TRA2 in females, these are proteins that affect the commitment proteins, and the presence of TRA2 allows the other commitment proteins to allow splicing at the female-specific splice site in the DSX messenger RNA. So that's one practical example of splicing. Let's look at it in a more abstract sense now. So you can have all sorts of different flavours of alternative splicing. So you can have alternative splicing where a cassette is totally excluded. So in this case, you can either have these 3 exons spliced together, or the alternative is you just have those 2 grey exons spliced together. You can have totally mutually exclusive exons. So in this case, in the upper variant, you've got this grey exon spliced to the pink exon, spliced to the grey exon. Or the alternative spliced version is the grey exon gets spliced to the blue exon, which gets spliced to the X exon. You can have intron retention, where the intron isn't cut out. You can have alternative 5' or 3' splice sites, so you can have cases where only part of the exon is removed. You can have alternative promoters, so allowing the first exon to differ. And you can have alternative splicing and polyadenylation sites, so you have different endpoints of the processed messenger RNA. And of course, you can have all of these in any sort of possible combination, which is how you can then, in some genes, lead to there being thousands of different splice variants. How's this controlled? There are various sequences nearest, that can be nearest to the various splicing signals that can either stimulate or inhibit splicing. And these sequences are involved in binding protein factors that can either activate or repress splicing. In addition to splicing, eukaryotic messenger RNAs are processed in 2 other ways. They have a 5' cap added, and they have a 3' polyadenylated tail. So at the 5' cap, then eukaryotic messenger RNAs are methylated. They have this 7-methylguanosine cap added, and this occurs very early in transcription. So this, as the messenger RNA is really short, this cap gets added on. And in particular genes, that cap can vary somewhat by differing in levels of O-methylation. There's a whole set of particular proteins that are involved in making this cap. You don't really need to know the exact details. The caps serve more than one function. So one of the things that they do is they help protect the messenger RNA from degradation. You may or may not recall, many lectures ago now, I discussed how bacterial messenger RNAs have very short lifetimes measured in minutes. Eukaryotic messenger RNAs have much longer lifetimes, half an hour, hours in length. And one reason is that they have this 5' cap that makes them much more stable. This 5' cap is also involved in enhancing translation of the messenger RNAs, and we'll look at that next lecture when we talk about translation. And it's also involved in signalling or that these messenger RNAs should be transported out of the nucleus into the cytoplasm. And it's also involved in affecting the splicing of the first exon. There's also, at the 3' end, there's a polyadenylated tail. So this is essentially a whole string of A base pairs. And this isn't made by RNA polymerase. There's not a whole string of T's in the gene that the RNA polymerase is recognising and making. Instead, there's a different enzyme called poly-A polymerase, which then adds this whole string of A's to the tail. And it can have up to 200 or 300 extra A residues added to the tail of the messenger RNA. And this poly-A tail serves similar functions, maybe almost identical functions, as the 5' cap. So it makes the RNA much more stable, so it has a much longer lifetime. It's important for translation of the messenger RNA. And it's also important for the efficient transport of the messenger RNA out of the nucleus into the cytoplasm. And this shows the basic mechanism of how that occurs. So here's our messenger RNA. The messenger RNA actually gets cut, and a bit of the 3' end of it gets cut off and thrown away and discarded. And then this poly-A polymerase comes along and starts adding A residues to that 3' cut site. And there are signals in the messenger RNA that signal to the cell where that cut site should occur and hence where polyadenylation should occur. And these signals are mostly A-rich region, about 20 nucleotides upstream of where that polyadenylation starts. If you make changes to that sequence, you can affect the level of polyadenylation, or the amount of A's that are added. This is a signal in mammals. Plant, yeast and various things also have signals, but the signals are a little bit different. But you can see also then this gives another way of a cell to control levels of expression. So if the stability of the messenger RNA is dependent on the poly-A tail and the length of the poly-A tail, if you have a polyadenylation signal that gives you a really long tail, that messenger RNA would be much more stable and hence last much longer and hence be able to be translated into more protein. If you've got a polyadenylation signal that's much weaker, there'll be far fewer A's added. That messenger RNA will have a much shorter lifespan and hence will make less protein. And there's a whole cellular machinery involved in the cleavage of this prior to polyadenylation, involving various proteins here, the details of which you don't need to know. And there's actually two separate phases to polyadenylation. There's an initiation phase, a cut occurs, and then there's a slow addition of a small number of about 10 or so A residues. And then once after that initial initiation step, poly-A polymerase then adds a large number of additional A's. There's a very dynamic process involved in these poly-A tails. Once the RNA gets into the cytoplasm, there are RNases that will chew back the poly-A tail, and some messenger RNAs have a sequence that signals for additional polyadenylation to occur, promoting extension of the tail. And this is another way then the cell can control the lifetime or the robustness of messenger RNA. And once the poly-A tail is completely removed, the messenger RNA is then targeted for destruction. So then, today's lecture we've looked at three things that are essentially occurring to messenger RNAs in eukaryotes. We've looked at splicing, we've looked at 5' capping, and we've looked at 3' polyadenylation. All of these things are actually need to be highly coordinated. So the cap gets added really early on, and the cap is actually usually important for the splicing of the first intron. And so you've got, and then the rest of RNA splicing occurs, and then the poly-A tail is sometimes important actually for the splicing of the last intron. And so you then wind up with this final process messenger RNA with all the introns spliced out with a 5' cap and a 3' poly-A tail, which can be hundreds of A residues in length. I've already said that splicing, maybe I haven't already said that, splicing begins while transcription is still occurring. Capping occurs early on when it's only about 30 nucleotides in size. Polyadenylation occurs while, or that first cut that signals where polyadenylation occurs, occurs while the messenger RNA is still being lengthened. Very briefly, there's potentially other things that happen to RNAs in the cell. So both ribosomal RNAs and tRNAs are processed in different ways. I'm not going to really talk about that today. In some organisms, messenger RNAs can undergo strange, weird post-transcriptional modifications. And this is usually in a handful of, I'll call them weird, lower eukaryotic parasites. And two of the stranger things that happen are transplicing and RNA editing. So in transplicing, and this is again, I think this occurs in one or two eukaryotic parasites, you actually get exons from entirely different genes spliced together. That doesn't happen in humans, doesn't happen in yeast, doesn't happen in plants, doesn't happen in most things. You can also have, in some lower eukaryotic parasites, you can have this process of RNA editing, where particular enzymes can come along and edit the sequence of the base pairs in the messenger RNA, then to make a different sequence again. Again, this is strange and weird and happens in only one or two organisms. Two other things that occur to RNA, so you can have post-transcriptional control of gene expression. So we've already discussed sort of how messenger RNA stability can lead to, if the messenger RNA is less able to be degraded, it will be able to make more protein. There's a whole set of machinery that can control messenger RNA stability and hence control the level of protein production. And then finally, there's a process known as RNA interference, RNAi, which in normal cells is involved in degrading foreign RNAs. But this has actually become a really important molecular tool in eukaryotes for molecular biologists, and Fire and Mallow won a Nobel Prize for this in the 90s. So this RNA interference occurs when cells encounter a double-stranded RNA, usually from a virus, and then there's an enzyme called DICER that will cut up that double-stranded RNA. Molecular biologists have been able to take advantage of this by, if they make RNAs that are complementary to an RNA of a particular gene, and so will base pair with it to make it double-stranded, that then signals to the cell, oh, this RNA is double-stranded, it must be a foreign RNA, I'll break it down. And so this actually allows you to greatly reduce, potentially totally eliminate expression from a particular gene through this process of RNA interference. All right, I'm out of time, so let's go on to the 30-second summary. So we've talked about messenger RNA processing. In the first half of the lecture, we talked about splicing, where eukaryotic genes are often interrupted by non-coding DNA or introns that have to be spliced out. This splicing mechanism involves forming a lasso-like intermediate of the intron. And the machinery responsible for splicing is the spliceosome, which consists of both RNAs and proteins, known as SNPs, and a set of additional proteins as well. We looked and saw how many eukaryotic genes undergo alternative splicing, giving quite different messenger RNAs that are then turned into different proteins. And we mentioned briefly that exons can contain particular sequence elements that control alternative splicing. And then in the last part of the lecture, we looked at how eukaryotic messenger RNAs undergo further processing, where they have a 5' protective cap added and a 3' poly-A tail added. Okay. Thanks, everyone.
Ian, what's that
So it's actually, I messed up what I was describing earlier. This is actually an intron which doesn't get cut out. So this intron's cut out here, but in this variant, the intron is actually... it might be that part of it has some active site that you then, if you remove it, it becomes non-active, or it might have some, you know, might be often it might be part of encoded part of the protein that has evolved in interacting with another protein. So if you splice that bit out, then it doesn't interact with. Yeah, so it gives you the ability to sort of tweak function of particular proteins, so they're either targeted differently or interact with other proteins differently, or totally non-functional.
Okay.